Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
A group of neuroscientists led by a University of Pittsburgh School of Medicine researcher developed a test to detect a novel marker of Alzheimer's disease neurodegeneration in a blood sample. A study on their results was published today in Brain. The biomarker, called brain-derived tau, or BD-tau, outperforms current blood diagnostic tests used to detect Alzheimer's-related neurodegeneration clinically. It is specific to Alzheimer's disease and correlates well with Alzheimer's neurodegeneration biomarkers in the cerebrospinal fluid, CSF. At present, diagnosing Alzheimer's disease requires neuroimaging, said senior author Thomas Karikari, Ph.D., assistant professor of psychiatry at Pitt. Those tests are expensive and take a long time to schedule, and a lot of patients, even in the U.S., don't have access to MRI and PET scanners. Accessibility is a major issue. Currently, to diagnose Alzheimer's disease, clinicians use guidelines set in 2011 by the National Institute on Aging and the Alzheimer's Association. The guidelines, called the ET-N framework, require detection of three distinct components of Alzheimer's pathology: the presence of amyloid plaques, tau tangles and neurodegeneration in the brain, either by imaging or by analyzing CSF samples. The development of simple tools detecting signs of Alzheimer's in the blood without compromising on quality is an important step toward improved accessibility, said Karikari. But the biggest hurdle in applying the ET-N framework to blood samples lies in the difficulty of detecting markers of neurodegeneration that are specific to the brain and aren't influenced by potentially misleading contaminants produced elsewhere in the body. For example, blood levels of neurofilament light, a protein marker of nerve cell damage, become elevated in Alzheimer's disease, Parkinson's and other dementias, rendering it less useful when trying to differentiate Alzheimer's disease from other neurodegenerative conditions. On the other hand, detecting total tau in the blood proved to be less informative than monitoring its levels in CSF. By applying their knowledge of molecular biology and biochemistry of tau proteins in different tissues, such as the brain, Karikari and his team, including scientists at the University of Gothenburg, Sweden, developed a technique to selectively detect BD tau while avoiding free-floating, big tau PR. To do that, they designed a special antibody that selectively binds to BD tau, making it easily detectable in the blood. They validated their assay across over 600 patient samples from 5 independent cohorts, including those from patients whose Alzheimer's disease diagnosis was confirmed after their deaths, as well as from patients with memory deficiencies indicative of early-stage Alzheimer's. The test showed that levels of BD tau detected in blood samples of Alzheimer's disease patients using the new assay matched with levels of tau in the CSF and reliably distinguished Alzheimer's from other neurodegenerative diseases. Levels of BD tau also correlated with the severity of amyloid plaques and tau tangles in the brain tissue confirmed via brain autopsy analyses. Scientists hope that monitoring blood levels of BD tau could improve clinical trial design and facilitate screening and enrollment of patients from populations that historically haven't been included in research cohorts. There is a huge need for diversity in clinical research, not just by skin color but also by socioeconomic background, said Karikari. To develop better drugs, trials need to enroll people from varied backgrounds and not just those who live close to academic medical centers. A blood test is cheaper, safer and easier to administer, and it can improve clinical confidence in diagnosing Alzheimer's and selecting participants for clinical trial and disease monitoring. Karikari and his team are planning to conduct large-scale clinical validation of blood BD-tau in a wide RA. Additionally, these studies will include older adults with no biological evidence of Alzheimer's disease as well as those at different stages of the disease. These projects are crucial to ensure that the biomarker results are generalizable to people from all backgrounds, and will pave the way to making BD-tau commercially available for widespread clinical and prognostic use. This research was supported by the Swedish Research Council, Vetenska Pradet, #2021-03244. The Alzheimer's Association, #AARF 21850325. The Bright Focus Foundation, #A2020812F. The International Society for Neurochemistry's Career Development Grant. The Swedish Alzheimer Foundation, Alzheimerfond. 2022, December 28. New biomarker test can detect Alzheimer's neurodegeneration in blood. Science Daily. Retrieved December 28, 2022 from ift.tt/odhypg University of Pittsburgh. Thanks for watching. Please subscribe my channel for more videos.